AgRP neurons and overeating. The brain circuit triggering uncontrollable late-night cravings. It's almost midnight. You ate dinner 3 hours ago. You're not hungry. You know you're not hungry. And yet something in your brain is screaming at you to eat, and it is winning. This is not a willpower failure. This is not weakness. This is a cluster of neurons deep in your hypothalamus, called AgRP neurons, doing exactly what they were designed to do. The problem is, they were designed for a world that no longer exists. And tonight, they just cost you 1,000 calories you didn't need. Here's the neuroscience of why, and what's actually happening inside your brain when late-night cravings feel physically inescapable. It's almost midnight. You ate dinner 3 hours ago. You're not hungry. You know you're not hungry. And yet something in your brain is screaming at you to eat, and it is winning. This is not a willpower failure. This is not weakness. This is a cluster of neurons deep in your hypothalamus, called AgRP neurons, doing exactly what they were designed to do. The problem is, they were designed for a world that no longer exists. And tonight, they just cost you 1,000 calories you didn't need. Here's the neuroscience of why, and what's actually happening inside your brain when late-night cravings feel physically inescapable. Section 1. What are AgRP neurons? Narrator, VO. Deep inside the hypothalamus, specifically in a region called the arcuate nucleus, sits one of the most powerful hunger control circuits in the mammalian brain. It contains two opposing populations of neurons that function like a biological push-pull system. On one side, POMC neurons, pro-opiomelanocortin neurons, which release alpha-melanocyte-stimulating hormone, alpha-MSH, activating MC4R receptors throughout the brain to suppress appetite and increase energy expenditure. These are your satiety neurons. On the other side, AgRP neurons, neurons co-expressing agouti-related peptide and neuropeptide Y, NPY, which do the opposite. They are your hunger neurons. And they are extraordinarily aggressive in their function. AgRP is not just a hunger signal. It is a competitive antagonist at the same MC4R receptors that POMC neurons activate, meaning it doesn't just promote hunger, it actively blocks satiety signaling simultaneously. It hits the accelerator and cuts the brake lines at the same time. NPY, co-released from the same neurons, acts on Y1 and Y5 receptors in the paraventricular nucleus and lateral hypothalamus, amplifying the orexigenic drive even further and triggering what researchers describe as a near-compulsive motivation to seek and consume food. When AgRP neurons fire, they don't suggest you eat. They demand it. Section 2. Why do they fire at night? Here's where the biology of late-night cravings becomes very specific. AgRP neuron activity is not constant across the day. It is circadian, rhythmically regulated by your internal biological clock, and in a pattern that, for most people, creates a neurological vulnerability window in the late evening hours. Three converging systems drive nocturnal AgRP activation. First, ghrelin. Ghrelin, the stomach-derived hunger peptide, is secreted in anticipatory pulses tied to habitual meal timing and circadian rhythm. Research published in the Journal of Clinical Endocrinology and Metabolism confirms that ghrelin levels rise significantly in the late evening in individuals with a history of nighttime eating, even when caloric intake during the day was adequate. Ghrelin directly activates AgRP neurons via GHSR1A receptors on their surface, one of the most potent activators of this circuit. When ghrelin rises at night, AgRP neurons respond with increased firing, and the hunger signal they generate is indistinguishable from genuine caloric deficit. Second, cortisol and stress. The HPA axis and circadian system are anatomically coupled, both regulated by the suprachiasmatic nucleus, SCN. Chronic psychological stress and disrupted sleep architecture elevate evening cortisol, which increases AgRP, NPY gene expression in the arcuate nucleus, effectively turning up the gain on the hunger circuit at exactly the time when it should be quieting down. A study published in Cell Metabolism demonstrated that glucocorticoid receptor activation in AgRP neurons directly increases their sensitivity to peripheral hunger signals, meaning stress doesn't just make you anxious, it makes your hunger neurons more reactive. Third, leptin resistance. Leptin, the adipose-derived satiety hormone, normally inhibits AgRP neurons through LEP-RB receptor signaling, activating JAK2, STAT3 pathways that suppress AgRP, NPY gene transcription. In individuals with chronic overeating, obesity, or metabolic dysfunction, leptin resistance develops at the hypothalamic level. AgRP neurons stop responding to leptin's inhibitory signal. The brake no longer works. The result? Elevated ghrelin, elevated cortisol, and non-functional leptin signaling, all converging on the same AgRP neurons at the same time of night. The circuit doesn't stand a chance of being quiet. Section 3. AgRP neurons bypass willpower. Here is the most important concept in this entire lecture, and the one that should permanently change how you think about late-night overeating. AgRP neurons do not work through conscious thought. They work around it. When AgRP neurons fire, they project to multiple downstream brain regions simultaneously, and the architecture of these projections explains exactly why late-night cravings feel biologically inescapable rather than merely tempting. Projection 1. The paraventricular nucleus, PVN, AgRP fibers densely innervate the PVN, the hypothalamic command center for energy regulation. By releasing AgRP at MC4R receptors here, they block anorexigenic signaling and drive food-seeking behavior at a subcortical level, before the prefrontal cortex has any input whatsoever. Projection 2. The lateral hypothalamus, LH, AgRP neurons project to the lateral hypothalamus, which contains orexin, hypocretin neurons, the arousal-promoting, reward-motivating circuits. AgRP activation through the LH promotes wakefulness and foraging behavior, exactly the nocturnal restlessness and food-seeking drive that characterizes nighttime eating. Projection 3. The nucleus accumbens, NAC, perhaps most critically for understanding why cravings feel compulsive, AgRP neurons modulate dopaminergic reward circuitry through projections to the nucleus accumbens. Hunger increases the salience of food cues. The sight, smell, or even thought of food triggers dopamine release that is disproportionately intense in a caloric deficit or pseudo-deficit state. Research published in Nature by Bentley et al. Demonstrated using optogenetics, the ability to switch specific neurons on and off with light, that artificial activation of AgRP neurons alone was sufficient to drive immediate, voracious eating behavior in sated mice within seconds. Not minutes, seconds. The same research showed that AgRP neuron activation generated a negative aversive signal, a form of discomfort or distress, that was only relieved by eating. The motivation to eat under AgRP activation was not pleasure-seeking. It was relief from an aversive internal state. This is the neuroscience of why late-night cravings feel like more than temptation, they feel like relief from something wrong. Because neurologically, that's exactly what they are. The prefrontal cortex, your seat of willpower and conscious decision-making, receives these signals downstream. By the time you're aware of the craving, the hypothalamic drive has already been running for minutes. Willpower is fighting a circuit that has a head start, more ancient wiring, and direct access to your motor systems. This is not a character flaw. It is a neurological asymmetry. Section 4. The nocturnal metabolic cost. The consequences of AgRP-driven nocturnal eating extend well beyond the caloric total. Your metabolism is not the same at 11 p.m. as it is at noon. It is circadian, and eating against your circadian biology compounds the metabolic harm in ways that go beyond simple energy balance. Insulin sensitivity and the circadian mismatch. Multiple studies, including a landmark paper published in Current Biology by Sutton et al., demonstrate that insulin sensitivity follows a circadian rhythm, peaking in the morning and declining significantly in the evening. The same carbohydrate load consumed at 8 a.m. produces a measurably lower glucose excursion than when consumed at 10 p.m. Late-night eating driven by AgRP activation therefore deposits calories into a metabolic environment with blunted insulin response, promoting hyperinsulinemia and preferential lipogenesis rather than glycogen storage. The clock-gene connection, the molecular clock within AgRP neurons themselves, governed by clock and BMAL1 transcription factors, regulates the rhythmic expression of AgRP and NPY peptides. Research has shown that disruption of this clock, through shift work, chronic sleep deprivation, or irregular eating patterns, dysregulates the circadian gating of AgRP neuron activity, allowing hunger circuits to fire at biologically inappropriate times. Essentially, every night you eat late and sleep late, you are rewiring the circadian expression pattern of your hunger neurons toward more nocturnal activity. The pattern compounds itself. Gut-brain feedback failure. Under normal circumstances, food consumption triggers GLP-1, PYY, and CCK release from intestinal L cells and I cells, gut hormones that travel via the vagus nerve and bloodstream to suppress AgRP neurons and terminate feeding. At night, the sensitivity of this gut-brain feedback axis is reduced, meaning more food is required to generate the same suppression of AgRP activity, further driving overconsumption before satiety is registered. Section 5. The science of breaking the circuit. Understanding the circuit is the first step. The second is using that understanding to intervene where the biology actually responds. Intervention 1. Time-restricted eating, TREA. The most evidence-supported behavioral intervention for reducing nocturnal AgRP activity is time-restricted eating, consolidating all caloric intake within an 8 to 10 hour window, aligned with the first half of the active day. A 2019 study published in Cell Metabolism by Wilkinson et al. Demonstrated that time-restricted eating, without caloric restriction, reduced body weight, blood pressure, and metabolic risk markers in a cohort of metabolic syndrome patients. The mechanism is partially hypothalamic. Consistent early feeding patterns entrain the circadian clock of AgRP neurons, reducing their late evening firing. When food is not consumed in the late evening habitually, the anticipatory ghrelin spike that activates AgRP neurons at night diminishes within 1 to 2 weeks. The circuit adjusts to the new pattern. Intervention 2. Sleep architecture restoration. Slow-wave deep sleep is the primary physiological context in which leptin sensitivity in AgRP neurons is restored. Research published in Sleep demonstrates that even one night of sleep deprivation reduces next-day leptin levels by up to 18% while elevating ghrelin, a hormonal shift that directly increases AgRP activity the following evening and drives documented increases in caloric intake. Prioritizing 7 to 9 hours of sleep, with consistent sleep and wake times to stabilize circadian cortisol patterns, directly reduces the HPA axis input that amplifies AgRP gain at night. Intervention 3. Protein timing. Research on macronutrient composition and hypothalamic signaling shows that protein is the most potent dietary suppressor of AgRP neuron activity, operating through PYY and GLP-1 release from the gut as well as direct amino acid sensing in the arcuate nucleus. Consuming adequate dietary protein at dinner, rather than concentrated carbohydrate loads, produces a more sustained suppression of AgRP firing in the post-meal window, reducing the reactivation of hunger neurons in the late evening. Intervention 4. GLP-1 receptor agonists. Pharmacologically, GLP-1 receptor agonists, including semaglutide and liraglutide, have demonstrated direct suppression of AgRP neuron activity in preclinical research. GLP-1 receptors expressed on AgRP neurons mediate inhibitory signaling that reduces both firing rate and AgRP, NPY peptide release. This is a significant part of the mechanism by which this class of drugs reduces appetite in a manner that many patients describe as qualitatively different from willpower, a reduction in the urgency and aversive quality of hunger itself. Late-night overeating is not a moral failure. It is the predictable output of a hunger circuit, AgRP neurons, that evolved to ensure survival in an environment of food scarcity, now operating in an environment of unlimited caloric availability and chronic circadian disruption. The circuit fires. Ghrelin activates it, cortisol amplifies it, leptin resistance removes the brake, dopaminergic reward makes eating the path of least resistance, and the prefrontal cortex, your willpower, arrives late to a fight that was already decided subcortically. Understanding this doesn't remove responsibility. It changes where intervention is most effective, not at the refrigerator at midnight, but in the meal timing, the sleep architecture, the protein composition, and the hormonal environment that determines whether those neurons fire at all. The circuit is not your enemy. It just needs to be managed, with biology, not blame. Subscribe if this level